So now to kick us off for Biomarkers in HD, please welcome Dr. Laura Byrne and Dr. Nicola Hobbs from the University College London to lead this session. Come on up.
As our speakers come up, they pose some questions in the Whova app to start some discussion. But as they're speaking, please put more questions in there, and we'll do some questions and stuff after their talks too.
All right, so actually we're making a small switch on you. I'm Tarina Martinez. I'm going to be presenting on the accepted endpoints in HD. The reason we changed the order is because biomarkers are endpoints. So I'm going to be providing an overview of the endpoints and then handing it over to Lauren and Nikki to go through the biomarkers. There's 2 pointers up here. I'm going to use the correct one. All right, my quick disclosure. I work for the Critical Path Institute. This was an organization that was formed as an independent nonprofit in 2005. It came out of the FDA's Critical Path Initiative. We get a good portion of our funding from the FDA. We're a public-private partnership. The rest of the funding comes from non-government sources. So we do get funding from the FDA. We work closely with the FDA, but very importantly, and this is the most important part of my disclaimer, I do not speak on behalf of the FDA. So when I talk about the regulatory considerations, that's because of the interactions that we have, but I'm certainly not the mouthpiece for the FDA. I mentioned that the Critical Path Institute was formed out of the Critical Path Initiative. We have several disease-focused consortia that work within this public-private partnership space. The Huntington's Disease Regulatory Science Consortium was founded in 2018. The overall goal of the consortium is to bring diverse stakeholders together within a neutral forum. So we bring drug developers, patient advocacy groups, nonprofit research organizations, clinician and academic researchers, regulators, really trying to bring the full perspective to the table so that we can outline a regulatory science strategy for Huntington's disease, de-risk the overall landscape for all of the involved stakeholders, make sure that we're incorporating patient centricity, and then advancing that in front of the regulators with early and often engagement. So we have iterated our next stage of our research plan to focus on earlier disease stages because there's a severe unmet need for drug development tools in that space. And you've heard other speakers before me in different days talk about the Huntington's disease integrated staging system. Dr. Sampaio talked about it this morning about how important of a research tool that it is. And we use this as an overarching framework to kind of organize and consolidate the work that we do within HD risk. So this is the schematic of how we kind of bring all of these diverse groups together. We have our engagements with regulators, we have our nonprofits. This includes HSG, CHDI, the Movement Disorder Society, and the Huntington's Disease Society of America. As I mentioned, we have a lot of clinician and academic researchers to bring that boots on the ground expertise. And then, of course, our pharma and biotech partners. We have 8 different partners currently. We all come together for frequent full coordinating committee meetings, and then we have broken out working group meetings. And then we use those mechanisms to evaluate the work that we're doing, identify our priorities, articulate unmet needs, et cetera. So then making a transition to regulatory decision-making and how we apply the considerations for regulatory decision-making to the work that we do and how this translates to what we're talking about today for endpoints and biomarkers. The considerations that help inform regulatory decision-making are the clinical context, the totality of evidence, the remaining uncertainty, and the unmet need. Clearly, each individual disease is going to have a different magnitude of each of these features, and it's up to the regulatory agency to apply their understanding and what the risk-benefit would be as they evaluate all of these 4 considerations in the risk-benefit calculus and also the safety efficacy calculus. So this is the overarching background that's applied to endpoints, to biomarkers, and interpreting whether or not the evidence and the unmet need and the level of uncertainty is balanced sufficiently for them to make a decision on endorsement or approval. So bringing this overall regulatory consideration framework to endpoints, what's really important is to identify the unmet need, determine clinical meaningfulness, articulate it in regulatory science terminology. It's not as easy as it sounds. Everyone in this room is struggling with the complexities of this space, and this is what makes advancing drug development for Huntington's disease as complex as it is. Part of that regulatory terminology, contextualization, and articulation needs to focus on some key elements, including the context of use. This is a statement that describes how a medical product development tool will be used and its purpose. And then there's also the concept of interest, the COI. This is a statement that describes a patient's state or experience that the assessment is intended to capture. These are going to be very specific, really narrow, and determine on a case-by-case basis, whether it's an endpoint, a biomarker, whether it's early stage, whether it's behavior, whether it's motor, whether it's something else. This has to be customized to the tool or endpoint. And then you also have to consider additional important factors, like the target population, bringing in that totality of evidence I talked about earlier. Always a question of data sufficiency, and in the rare disease space, the data sufficiency is always a painful, rate-limiting step. And then there's an important distinction between qualification and endorsement. So when we talk about the full approval of an endpoint, a biomarker, a drug, that's full qualification. Often this can take massive amounts of resources and several years. But then there are also earlier versions of regulatory endorsement that can be obtained. For example, you can get a letter of support that indicates from the regulatory agencies their endorsement of something. Maybe that's using a biomarker for exploratory purposes or trying a new endpoint in a certain narrow context. So there are opportunities to get endorsement that's more upstream and less time intensive. It doesn't mean that the standards are lower or the rigor is compromised. It's just another way to try to think creatively and move more efficiently in the space. So then another consideration for endpoints and other important drug development tools in Huntington's disease is real-world data and real-world evidence. So this is data and evidence that is obtained not in clinical trials. This could be through some of the clinical studies that we've heard a lot about from the updates with HSG. Obviously, Enroll is an example of a clinical study that is not a trial, but is gathering really high-quality information and data. So that is obtaining the data in the real world. At a certain point, that data is rich enough, consistent, high-powered, longitudinal, or whatever the criteria need to be to get it considered as real-world evidence. And then it can be used to inform regulatory decision-making. And this is kind of how we can develop the critical mass of data and evidence for the regulators to then entertain approving a biomarker or an endpoint in a trial, even if it didn't come from the clinical trial landscape itself. So I realize this is a lot of jargon on the regulatory bit. I hope no one's in a coma from the snacks. So I'm hoping to take what is a very important feature in this drug development space that can sometimes be mystified or inaccessible because of the kind of black box that we think about when we look at the regulatory considerations. But clearly they're very important, and it's something that we bring to the projects that we work on at HT Risk. So all of this, the context of use, concept of interest, all of these questions around totality of evidence are applied to endpoints and also applied to biomarkers. Biomarkers are endpoints. I'm not going to dig into biomarkers at depth because that's what Lauren and Nikki will talk about shortly. But when you're thinking about the same considerations of context of use, concept of interest, that has to be applied to the biomarkers as well. And then how that biomarker is applied is going to determine the amount of evidentiary data that's required, and that will be determined by the use of the biomarker, whether it's going to be used for safety or prognostic or response. That requires different levels of data and a different consideration for context of use and concept of interest. So now moving to the clinical endpoints, I want to just kind of take a moment for definitions and hierarchy here. So the clinical endpoints are specific outcomes or events that are measured objectively and quantitatively in clinical trials to determine the safety and effectiveness of a treatment. And they need to be validated beforehand. You can't just sneak it in and then come with your data and voila, claim that it's validated. There's a lot of upstream work that has to be done. And then it's important because clinical trials often will contain multiple endpoints. So they will typically involve several different endpoints that are kind of hierarchically arranged around primary, secondary, exploratory. So primary endpoints are the most important outcomes and are used to determine the study's overall success. Secondary endpoints provide additional information to help researchers understand the treatment's effects, and of course, that will be evaluated by the regulators. Exploratory endpoints are used to interrogate new research questions or test new hypotheses and to get more data for future application and study. And they are distinct from the primary and secondary endpoints because they're answering specific research questions and provide supportive data. Dr. Sampaio talked about surrogate endpoints earlier this morning. I'm not going to talk much about that because she did such an excellent job covering the importance, the complication, how it applies in Huntington's disease. But just want to remind you, or if you weren't here in the morning, the surrogate endpoint is an endpoint or an outcome or event that is used as a substitute for the actual clinical outcome. It needs to demonstrate clinical benefit or be reasonably likely to demonstrate clinical benefit, and it also needs to have treatment response. So that's a high bar, but if you can show that and get accelerated approval, then that does move things along faster. So then the FDA has many, many guidances that they have pulled together for the community to really understand their thinking and to telegraph what they think are the important criteria and how things are evolving in the space. And so one of the things that informs the current approach that the FDA brings to how they evaluate endpoints is patient-focused drug development, which is a systematic approach to help ensure that patients' experiences, perspectives, needs, and priorities are captured and meaningfully incorporated into the drug development and evaluation. So this means it needs to be not just a late to the party sort of consideration, but something that's integrated at all steps and high upstream in the process. I want to emphasize again that endpoints can be clinical. They need, in the context of clinical trials, these are clinical endpoints, but they can be either a clinical outcome assessment or a COA, that is a measurement to describe how a patient feels, functions, or survives, or they can be a biomarker. So the biomarker measures a substance that indicates normal or abnormal condition, process, disease, or response to treatment. And again, Nikki and Lauren will be talking about the biomarkers in just a moment. But then talking about the different types of clinical outcome assessments, that can have a wide variety of methodology and data depending on how they're designed, the questions they're asking, what they're measuring, coming back to that concept of use, concept of context of use, concept of interest that I was speaking about earlier. So you can have patient-reported outcomes. Obviously, this one's pretty self-explanatory. This could be symptom diaries. It could be surveys, questionnaires. You can also have clinician-reported outcomes. This is going to be clearly information and data that's obtained from the clinician or a trained medical provider. You can have observational reported outcomes. These can come into play in situations where caregivers provide the information, whether by survey or other mechanism. It can also be used in situations where there's juvenile disease and a parent will be providing that information. There's also performance outcome, and then digital health technologies are another tool that can be used. And what's distinct about DHTs is that depending on how they're applied, and again, the complexity of that context of use and concept of interest, they can either be a clinical outcome assessment or a biomarker, again, just to provide another element of complication to the process. But it depends on how they're developed and what data they're measuring for that to be determined. And the FDA has distinct pathways to evaluate the qualification of COAs and biomarkers. I'm not going to spend time or detail here, but it's important when we look at the endpoint space. And I mentioned earlier, there can be that hierarchy of the primary, secondary, exploratory, surrogate. Since there are multiple endpoints in the clinical trials, there need to be considerations for how they're applied, whether they're co-primary endpoints, whether they're multiple endpoints. Each time there's going to be some give and take of additional complexity or benefit, depending on how the endpoints are applied and how they're analyzed and what the data indicates. There has to be care and attention to the analysis plan and make sure that, bias or errors and other complications of the data analysis are addressed, because sometimes when you add additional endpoints, clearly you're bringing more complexity, and that can change the statistical analysis plan, et cetera. And then, I'm not going to get into the detail here. This is why the composite endpoints has multiple bullets and microprint. But the composite endpoint has multiple features that are, or domains that are included. So there's often benefit to that, especially in diseases that are heterogeneous or complex, because it might give you an opportunity to capture multiple features. But then it's also possible that those different domains and features have different levels of clinical meaningfulness, or maybe they have different impact in earlier versus middle versus later stages. So it can provide sensitivity and give you more to look at and understand, but it can also provide complexity, and you have to be careful in how you approach that. And then the last kind of endpoint is the multi-component endpoint, and this is when you're combining two or more components. But again, you have to be careful on, what you're measuring, what direction they're moving, how you're interpreting the data, and then bringing that to the regulatory consideration. All of these breakdowns came from the FDA multiple endpoint snapshot. They have a whole guidance document from October 2022 for industry on how to apply multiple endpoints in clinical trials. So now moving to the approved primary endpoints for HD. There are some approved primary clinical endpoints for HD, but there are not currently any approved biomarkers to be used as a primary endpoint. That's a very active discovery space, and you'll hear about that in just a moment. But the composite unified Huntington's rating scale, which we've all heard about, many of the clinical trial readouts from earlier today and yesterday, the CU-HDRS is an approved primary endpoint. This was developed by HSG to help quantify and categorize HD symptoms and severity. The FDA has approved it as a primary endpoint. And then importantly, I want to point out that the CU-HDRS is one of those composite endpoints that I spoke about earlier. It has multiple features and domains that are being measured. And actually, some of those subdomains have been parsed out, like TFC, total functional capacity, total motor score, TMS, and those have individually been approved for use as primary endpoints. So just want to explain that those are actually part of that UHDRS, but they can be individualized. If what you're really looking at is specifically motor, then you can just use TMS, for example. And then there's also the HD CAB. This is the HD Cognitive Assessment Battery. This is also a composite endpoint, and it's been approved as a primary outcome. So I want to point out, coming back to the HDISS, the staging system. Again, I want to remind you, Dr. Sampaio emphasized that this is a research tool. It is absolutely an important research tool, but we're using it as an overarching framework to understand where we are in the endpoint space, in the biomarker space, in the disease stages that lack good endpoints. Because if you kind of overlay, mentally picture it, over the different stages and the different progression bars of the staging system, each one of those is going to have its own concept of use, context of interest. I mix those up. Concept of interest, context of use, and the target population, because you're looking at different discrete stages. And so if you're really using the specificity of what's going on biologically or pathologically at those individual stages, that's going to inform and specify what biomarker you're looking at, or if you have a biomarker yet. If you think about it, in the early disease stages that aren't, where you don't have symptoms, you don't have deficits in function, you can't be using a clinician-reported outcome, because a clinician-reported outcome is capturing deficit in function, symptoms. So if you're shifting to the left side of this graph, then you can't be looking at a clinician-reported outcome. You can't be looking at a clinical scale. They're not in play yet. There is a paradox understanding clinical meaningfulness for a lack of symptoms and a lack of deficit of function. Clearly, if you ask individuals who are at risk for developing or progressing in Huntington's disease, they would tell you it's very meaningful to not progress. But how do you measure nothing? And how do you quantify that? I mean, this is what the field is wrestling with, and this is why we need things like biomarkers that you can tie to the pathology downstream and tie to the biology of the understood mechanism, and show that even if you have a lack of symptoms, you're measuring something meaningful, and then you can quantify and see whether or not it's responding to treatment. So that's the big picture. This is just taking that integrated staging system and overlaying it with some of those approved endpoints that I talked about earlier, looking at TMS, SDMT, TFC. You can see that there's complexity to this already simplified graph because it shows that there's not like, the clear cutoffs between the stages that we showed in the earlier graph for just visualization purposes, but you can see that different endpoints are going to have different impacts or be present or not present in different stages of the disease. And so this is why we need multiple endpoints. This is why we need improved endpoints that have more sensitivity. This is why we need biomarkers to add sensitivity and measurement in the earlier stages, and that's what you'll hear about next. So then I kind of talked about the regulatory considerations around endpoints, the approved endpoints, the need for additional endpoints, and there is a lot going on. This is an active space. I do not even pretend that this is an all-inclusive list, but I just wanted to capture for you some additional endpoints that have been used for secondary or exploratory endpoints in HD trials. I'm not going to go into them in detail here, but for example, the clinical global impression of change and other measurements for motor, digital assessments have been used, but they need to be further evaluated, and additional data needs to be considered before they're approved for primary endpoints. And then there are a lot of measures and assessments that are in development for potential use. I've listed a few of those here. And again, there's a lot of activity in this space, but we have to think about what we're measuring, how we're measuring it, validate the measurement, work with the regulators, et cetera. So then just to wrap up my part and hand it off to Lauren, I just wanted to kind of bring you back to the overall impact. Like, go from the granular, I started out general, went granular, want to come back to a little more general, and really just emphasize the impact that these drug development tools can have. So we talked about the HD-ISS as a research tool, but we're using it now to better understand how we can improve the endpoints and the regulatory considerations across the disease spectrum. This can help in clinical trial enrichment, and we're using it now to help inform potentially prevention trials. We can use clinical trial simulation platforms to help fill in the gaps and bridge the knowledge gaps for the gaps that we have. The neuroimaging and fluid biomarkers, which you'll hear about immediately next, are necessary to improve the ability to detect disease onset, and they need to sensitively measure progression. And then when we talked about the endpoints and other outcome measures, these can improve the ability to detect onset and then changes in meaningful symptoms. So I just wanted to kind of leave you with that as the stage that you can now add the additional information and details that Lauren and Nikki will tell you about for fluid and imaging biomarkers. With that, I want to thank, this is my village. My village is my consortium here, listed on the screen. It takes this whole group coming together to really help us make progress in the space. And so I just really want to thank everybody. I don't have time, obviously, to go into the individual, but the collective effort and genius that's in the space is what moves us forward. And with that, I will hand it off to Lauren.
Clarification that is needed in the slide that you presented, saying that there was approved primary endpoints. Okay. Maybe, is it possible to go to that slide? If not, the thing is, I don't know if this was what you meant, but if that list is not approved primary endpoints, because single-digit modality test has never been approved, the HD CAB is a matter of big contentious. So HD CAB, so far, many companies would like that, but so far, HD CAB has been refused systematically by FDA. And the single-digit modality test, although it has been approved in the context of multiple sclerosis, it has never been used as primary endpoint, and therefore, let's say, supported by FDA. So I think the approved primary
Yeah.
endpoints title is a bit misleading.
And thank you for the clarification, Christina, and it certainly is not my intention to be misleading. There's also, as I mentioned, all endpoints have to be validated before they are used in the clinical trials, and sometimes there are clinical trials that are starting to enroll or have not yet begun that list different endpoints as their intentional primary outcome. So there is, I just want to apologize for any of the misleading component there.
All right, fluid biomarkers, my favorite. So I'm going to get stuck in. You can have lots of different types of fluids that we can extract from humans, anything from saliva, urine, blood, and probably more popular now in the last few years in Huntington's disease, CSF, or spinal fluid. We're also looking at other fluids, but for the purpose of this talk, I'm going to focus mostly on blood and CSF. One example, I want to go back to 2017, where we had a very exciting time when we were able to show for the first time in HSG patients that you could target the Huntington protein and lower it in a dose-dependent fashion. So this is the first proof of concept that we could actually, in humans, target the pathogenic species in HD. But what some people don't know is that our ability to be able to find that target engagement and proof of concept was based on this tool, being able to measure the mutant Huntington protein in CSF, which was actually published months before that trial even started. So without that tool, you can't actually get that target engagement and make those decisions, no-go or no-go decisions for therapeutic development. One thing I would also highlight here is kind of the difficulties in being able to even make this assay, which we've heard earlier today. There are a lot of issues with the measuring mutant Huntington in CSF. It's a intracellular protein in the most part. It's very low concentrations in the CSF. It's taken a platform, what we call ultrasensitive immunoassay technology called, this is the old platform, but it's now in the SMC Pro by Merck, using these antibody pairs. So I just want to highlight here as well, measuring particularly biofluid biomarkers, not all assays are the same, not all targets are the same. So it's why I question people on kind of what tool did you use when you're saying you've increased NFL or lowered Huntington. It's, these are the things that I nerd out on, but yeah. So Tarina mentioned kind of all of these need their actual use and context of use and category. So it's not enough to say something's a biomarker of HD. What is it going to be used for? Is it disease progression? Is it target engagement? And we all know that Huntington is going to be super important as a pharmacodynamic for all of the different therapies looking at Huntington lowering. So, changing from Huntington to my favorite protein, neurofilament light, or NFL, which we've heard a lot about already. For those who don't know, neurofilament light is a protein found in all neurons. It's a cytoskeletal protein that helps neurons hold their caliber and shape, which is ultimately important for their function. And when there is injury to neurons, we see that it gets released into CSF, and now after ultrasensitive immunoassay technology like Simoa, we can measure it very robustly in blood. And it seems to be a kind of putative marker of neuronal injury, so not disease-specific, but disease process-specific. So what we found over the years in Huntington's disease, first in the TRACK-HD cohort, we showed for the first time a biomarker that was elevated this far from onset, and that was over between 15 and 10 years from onset. It also had this really striking relationship with CAG repeats. A single measurement at baseline could predict subsequent atrophy, and also it was predictive of those who progressed to motor onset. In the HG-CSF cohort with Ed Wild, we looked at then head-to-head mutant Huntington in CSF and blood in CSF NFL, where we were to show that cross-sectionally that NFL had stronger associations with the clinical scores, with brain volume, and using some modeling methods, trying to show a sequence of event of where biomarkers change on the timeline of HD. These biofluid biomarkers seem to change earlier. And longitudinally, you can see kind of highlighting kind of some of the issues with mutant huntingtin, where you see even in manifest patients, some people be below the lower levels of quantification, or the limits of quantification, which are the limits of being able to technically and validly measure something reliably. A lot of people are under those limits, which is a huge issue currently in trials, trying to use this as a pharmacodynamic readout. But here we could see the NFL longitudinally changing very early in the disease progression. And we had some insights into kind of blood NFL being one of the strongest predictors even before things like disease burden score or CAP score. And NFL is obviously very important to a lot of diseases. It's been mentioned before, again earlier by Christina, talking about accelerated approval in the ALS trial of tofersen. But it really highlights why we need to really understand what NFL is doing in HD and what the real context of use of NFL will be in our disease. Is it going to be a surrogate endpoint that when we treat someone, we're going to see a change in NFL and that will predict the clinical benefit, as you've seen in kind of the Cinderella stories of SMA, where you see drastic drops in NFL that predict the clinical benefit and very obvious clinical benefit. But unfortunately in HD, we have yet to find a drug that has changed and modified disease progression, as already mentioned today. But what is going on with NFL? How do we really interpret these changes? So for example, in a traumatic injury, so this is in healthy boxers that have went through concussion, and you can see these rises in NFL, which kind of translate to up to 25-fold change, or around 725% change, and it can take up to 6-9 months to get that back to normal levels, just to kind of give you a context of how NFL reacts in a non-disease situation. Then thinking of the tofersen and ALS trial, where you did see substantial change in NFL, and much more subtle and evidence of the clinical benefit, as Christina highlighted earlier. But in other diseases like Alzheimer's, we don't see a change in NFL, but we do see clinical benefit. And in other cases, it might take several years before we see the lowering of NFL. So it's just something we have to really consider when we're trying to decide how NFL is going to change in the context of benefit in Huntington's disease. So then when we start thinking about our situation of wanting to move earlier in disease and trying to treat people potentially in this stage 0-1 stage of a person with Huntington's disease. But really, these stage 0 to 1 can span over 20 to 30 years of someone's life. So we need to have ways of stratifying within those groups. So Jodie Corey Bloom's group have done some nice work looking at the HDISS using a PIN score, approximated stages for stage 0 and 1. Showing that NFL is increasing at those stages, and some unpublished work in our own centre looking at CSF NFL suggests that it could be changing in the stage 0. But also NFL has clearly an important role as a safety biomarker. We've seen a lot on the tofersen trial. So what really happened there? So a lot of you've seen this data before. There was no clinical benefit, and actually people that were on the drug got worse in a dose-dependent fashion. And Peter presented a lot of the biomarker data. This is an older version of that slides that he presented earlier, but we see exposure and Huntington lowering that can't be disentangled because they're both related. But we see the increases in biomarkers, particularly this transient NFL spike. But one thing, done the wrong one. I think one important factor of that study is that NFL went up and back down after continued Huntington lowering. We also saw the evidence from the open label extension of the earlier phases of that trial where they doing the 4-week regimen, and you see very clear evidence of increase of white cells and protein, which is indication of neuroinflammation. And this is from the supplementary of the phase 1 trial, where we saw these initial spikes, transient spikes of NFL, which really translated to around 54% increases, with the kind of maximal up to 240%. PTC have presented some really exciting results, able to show the dose-dependent reductions in mutant huntingtin. And then when we look at their NFL, I think it's really important to note that they are not seeing the spike. And they didn't mention, but I think the range here that they've included is actually based on another drug, Pranaplam, which did show NFL spikes, which you can see here, although this is the blood data, but they had quite marked NFL increases, which also translated to adverse events such as neuropathy. I think this is really important to highlight, that this is the first evidence to suggest that actually lowering huntingtin systematically, wild-type huntingtin, does not inevitably mean that we're going to damage healthy neurons. So when we move to WAVE, also some really exciting results. It's been a positive year in HSG research. They showed the dose-dependent and selective lowering of huntingtin. But they also highlighted this earlier, the increase in NFL, which I wanted to put into a bit more perspective in terms of comparing with the same amount of NFL increases in other trials. So you can see their dosing regimen here, and just putting them into context in terms of the tominersen changes. I've added the 4-weekly level here, and this level is around 300%. So I don't think these are increases that should be ignored, and I know that WAVE are taking this seriously and are considering their dosing regimen in the next phase and planning their next trials, but I think it's just important for everybody to get a perspective on what these may mean. Then when we look at a trial with brain surgery, so things get a bit more complicated because you get there's a lot of exciting results from the Unicure trial that were presented earlier, just to remind people of the data where they showed some suggestive improvement in the CU-HDRS. But when we think of NFL, so as expected, we see a very sharp increase, about 400% increase in NFL, because you're actually drilling into someone's head and causing brain damage. But this is obviously transient, and when we zoom in later, after up to 24 months, there is evidence to say that we're going below baseline, and when they compare this to the natural history data that was matched to the cohort, they have evidence to suggest that this is positive. So, and just a summary of kind of those trials and what we're seeing in terms of Huntington and NFL. Lots of other companies are coming through, whether they're huntingtin lowering modalities or gene therapies, and also importantly to think about is those companies that are starting to look at genetic modifiers and DNA repair pathway. And this is kind of a big topic for biomarkers, because not only will they need target engagement markers for if they're trying to lower any of these or increase any of these to show that they're hitting their targets, but we also don't have a very robust measure for in vivo and timely, as Christina mentioned this morning, to measure somatic instability. I've took some snapshots of Darren Monkton's slides from CHI last year, where he was starting to talk about the potential of being able to measure somatic instability in blood, where we can see kind of steeper slopes and higher CAG repeats. But as we know in the standard clinic and the more common people that we see in clinic have CAG repeats from kind of 40 to 45 most commonly, and we see much smaller slopes, so perhaps not feasible to measure within the time frame of a clinical trial. But there are works to potentially use repeated measures to increase signal to noise and other bioinformatic options to improve this further, which Darren's working on. Another they did warn me about the button that I shouldn't touch. I clearly listened very hard. There we go. So other avenues that people are approaching is to perhaps look at different compartments within biofluids, like extracellular vesicles, which are basically little pockets of cells that break off that may contain DNA, RNA, or protein. And we could potentially collect these from CSF, which would hopefully be more brain enriched, or enriched blood biomarkers for markers for brain proteins. And hopefully this may be an avenue in the future to look at somatic instability. One marker I'd wanted to highlight, which was mentioned a bit, is proenkephalin. So some of the issues and maybe criticisms of neurofilament light is the fact that it's not disease specific. It is specific to a process. We could really benefit from a complementary marker that is actually specific to HD, that represents something very specific to the biology, for example, the striatal pathology that we see. Proenkephalin is highly expressed in medium spiny neurons that are most vulnerable in HD, and using multiple papers have presented using mass spec methods, being able to target the similar peptide with success and shown changes in HD, with showing lowering from the pre-manifest stage in two sample collections, as well as showing the disease specificity where only HD has the lowering even compared to Parkinson's and Alzheimer's. And again, another study with Nick Cairns and Michael Hayden's team showing that kind of early pre-manifest and early HD changes. So it suggests that this is something that might be changing earlier and something that we look forward to seeing more data. And Sarah Tabrizi presented some exciting data at HDN, and I know Nikki's going to mention it a bit in her talk, and hopefully maybe Peg might be interested in looking at something like this and change HD in these really far from onset cohorts. So as I finish, I want to highlight thanks to Ed Wild, because I borrowed some of his slides from HDN and adapted them. That's how he looks all the time. And a big thanks to my team. So I haven't had a lot of time to talk about the work that I do, but we're particularly interested in really building that data set of NFL across the HGISS, so working with a lot of people in the field to get as much blood data on NFL and harmonize it so we can get cutoffs for all the HGISS stages, as well as developing remote blood collection methods for NFL to hopefully expand our reach and be able to reduce the burden of in-person assessments, increase our capability of doing frequent sampling, et cetera. So lots of people involved in our work, but most importantly, I always thank the family members and patients that this is some of the social media from our at-home collections where people are collecting their own blood and sending it in the post every 2 months from the young adult study, so they're incredible. And I look forward to questions later.
Good afternoon. I'd like to thank the organisers for the opportunity to speak today. It's a real pleasure to be here. So I'm going to talk about imaging biomarkers in HD. I'll start off with a really brief recap on the prognostic value of imaging biomarkers and what we've learned from natural history data. And they're going to move on and talk a little bit about the current application of imaging in interventional studies and talk about some of the challenges as I see them, because I think maybe we're not discussing the challenges enough at the moment. And I'll show a few examples. And then finally, I'm going to finish off looking ahead to potential preventative trials, and I'll share some new data with you from the HD young adult study, which we're currently working on at UCL in London. So we all know there are many different imaging modalities that are currently being studied in HD research for things that probe structure and function in the HD brain, and also ways of tracking disease progression. By far the most mature and advanced is volumetric MRI, so this is going to be the focus of my talk today. So volumetric MRI already plays a role in HD trials, so cross-sectionally in terms of participant selection, and then longitudinally in terms of secondary and exploratory endpoints. We have central imaging facilities which are well positioned to standardize data collection for central quality control and image analysis. And this is really vital to the integrity of imaging data in trials. There are also harmonized acquisition sequences available, although it is worth noting that they weren't specifically developed for use in HD. You can get high-quality scans in 5 to 8 minutes, so the participant burden is relatively low. In reality, we tend to get back-to-back T1 images, and that increases the probability of a really good quality scan for analysis. Here I've just shown an example of the qualification scans from the 4 sites in the Track HD study. It just gives you an idea of what well-standardized high-quality data can look like. So we've got nice contrast, we've got full brain coverage, and we've got minimal artifacts. So we know that it's feasible to acquire volumetric MRI in clinical trial settings, but we also have a lot of data from natural history studies that really supports its use. And some of the important ones, or the bigger ones, we've got Predict HD, Track HD, and Image HD, which have really been informative. So in terms of the prognostic value of volumetric MRI biomarkers, so we've all seen images like this before, and we know that cross-sectional brain volumes are reduced in Huntington's disease prior to clinical motor onset. In the middle, we can see this scan from a pre-manifest individual. We can already see enlargement of the lateral ventricles, reduction in size of the caudate, and actually, in early manifest diseases, these changes are more pronounced and they're more diffuse. So we can look at this on a group level. Top left in figure A, we can see data from the Track HD study. So we see a stepwise decrease in caudate volume from a pre-manifest A group through to early manifest disease. And although there is a stepwise decrease, it's also important to note that there is a lot of overlap in volumes between those different groups. Figure B, we can see that caudate volume is associated with disease burden score, and there's a very similar relationship for the putamen. Figure C, that's data from the Predict study. We can see the relationship, the linear relationship with putamen volume and actual years to diagnosis. Figure D, this is a spatially unbiased analysis, and it again supporting the idea that volumetric reductions are really starting off in the caudate and putamen and become progressively more widespread as the disease progresses. We also know that cross-sectional regional brain volumes are associated with clinical outcomes. So on the left-hand side, we can see that reduced volume in the caudate and putamen is associated with impairment in a tapping task. And on the right-hand side, we have data from Predict, again showing that reduced volume in the caudate and putamen is associated with performance in several motor tasks and also poorer performance in several cognitive measures, including the SDMT, which was mentioned earlier. But when it comes to thinking of biomarkers of disease progression, I think volumetric MRI really stands out. It's because it has robust longitudinal change characteristics, and this is consistent in different observational studies. So on this slide, I've shown how caudate, putamen, and whole brain volume change with time at different stages of the disease. So we can see in green the control group. Blue is the pre-HDA group, which were over 10.8 years to predicted onset. Then purple, we have a pre-HDB group closer to predicted clinical onset. Orange is the Soltion-Fan stage 1, so that's manifest disease, and red is stage 2. So for caudate and putamen, in particular, we can see this linear decrease in volume over time, and we can see really good group separation, and it's sensitive even between controls in this early pre-manifest group. Whole brain volume is a similar story, but it's much less sensitive in pre-manifest. disease. So we can also look at longitudinal effect sizes from natural history studies and use this to infer information on sample size requirements for clinical trials. So we see again and again that volumetric MRI effect sizes are larger than for clinical instruments. And this graph here is just an example in manifest HD. So on the y-axis, it's longitudinal effect size, and then on the x-axis, we see different brain regions, and then also the TMS. Green is the Paddington study, which was collected data over different short intervals, and blue is the TRACK-HD study. So if we look at TRACK-HD data over 24 months as an example, we say caudate longitudinal effect size of 2, compared with TMS, which is about 0.8. We can then see how that we can derive idea of effect size of sort of sample sizes per group, assuming an effect size in this example of about 40%. And you can see theoretically how the numbers are much smaller for the caudate. So it clearly has huge potential in terms of helping to provide evidence for disease modification in clinical trials. There's also some evidence that change in volumetric MRI measures predicts change in clinical outcomes. So, for example, in the TRACK-HD study, we showed that regional volume change predicted change in TFC over 36 months in early HD, and this was particularly notable in the caudate atrophy. The IMAGE-HD study showed that caudate volume change was associated with change in reaction time during cognitive tasks over 30 months. But I think there is a need to better understand or to have a more complete understanding of how longitudinal change in volumetric MRI measures relates to change in clinical measures on the timescale of a clinical trial. And I think that will aid us in terms of interpreting clinical trial data. So I'm going to shift gears a little bit now and think about application of imaging to clinical trials. Firstly, cross-sectional volumetric MRI for participant selection. So based on the natural history data, caudate volume and putamen volume were selected as landmark biomarkers of pathogenesis for the HD-ISS staging. So basically, if either your putamen or your caudate falls below a set threshold and your CAG is greater than or equal to 40, you'll be categorized as HD-ISS stage 1. I think it's really important to note that the published cutoffs are based on using FreeSurfer version 6. And actually, the software and the version are really important. If you use a different type of software, or even if you use FreeSurfer version 7.4, you'll get a different volume for the caudate putamen, and that may well change the staging. So that is definitely something to keep in mind. It's a subtlety, but it's a technical subtlety, but it's very important. In terms of FreeSurfer version 6, it's good in that it's freely available, it's fully automated, and it's well documented. But we also need to be aware that, as in lots of automated segmentations, the segmentations are not perfect. And this image here shows an example of a common problem with FreeSurfer in terms of segmenting the putamen, in that the classroom is often included. And these are actually very good quality scans, and so there are also situations that could be much worse than that. So I think if you're using caudate and putamen volume measured with FreeSurfer in a clinical trial, it's really important to have a predefined plan for identifying and managing suboptimal automated segmentations. Oh, this is my dream. So I would really like there to be some sort of lookup table, so actually you could use different software or up-to-date, the latest release of software, and there could be different cutoffs, so basically a bit of a data dictionary. That would be lovely. So let's move on to another example, thinking about enrichment biomarkers for gene therapy trials. So this is just an example from the Unicure trial, where minimum requirements for putamen and caudate size. And the idea is to promote both safe and effective surgery. I believe there currently isn't a consensus on these volumes, so it'll be interesting to see how this sort of discussion evolves. Again, there's the measurement considerations, thinking about software, endpoint quality control, and also there's the additional thought of whether neurosurgery causes issues for the other volumetric MRI endpoints, and that's something I'm going to come to in a moment. So let's move on to longitudinal volumetric MRI biomarkers for assessing the effects of intervention on atrophy. So again, based on natural history data, we have percent volume change from baseline for selected brain regions. So number one, regions differentially impacted in HD. These are by far the most sensitive biomarkers and can be used in the earlier stages. So caudate has been used as a secondary endpoint in clinical trials, such as the Legato HD study, and also putamen volume. They're sort of considered the top two, really, for sensitivity. There's also global measures, and these have the advantages of encompassing the global effects of the treatment on the brain, but they do lack sensitivity. So things like whole brain volume, lateral ventricular volume, and grey and white matter volume, they're often included as exploratory endpoints in trials. There's also a move more recently to consider also including a region minimally impacted in HD, such as the hippocampus. And the idea of this is that it may aid interpretation. So when you're trying to disentangle the idea of how the therapy has modified disease progression, or whether you think there might be some confounding effects on your volumetric MRI, having a sort of a global idea of what's happening may be helpful. This is a little bit of a bugbear of mine. So different image analysis tools have different sensitivity, reliability, and accuracy. So it's not just what you measure, it's how you measure it that's really important. And there isn't a consensus in the field about the best measurement tools, but I think as a sort of a bare minimum, you should use something that's already been shown to be effective in a natural history study over the interval that you're looking at in your trial. Okay, so I want to move on to challenges now. So there are lots of challenges, but I've picked my top three. Number one, interpretation. So in a natural history study, volumetric MRI behaves really nicely. So you have your HD group and a control group, and the volume changes can really be attributed to atrophy, so loss of neurons and the connections. Sometimes in an interventional study, interpretation is more complex. For example, you might have reductions in inflammation, and this may look like an increase of atrophy. And this is something that has been experienced in some MS trials. It's called pseudoatrophy, and actually you can mitigate against it in terms of your clinical trial design, something to bear in mind. Another important one is how treatment side effects may confound therapeutic effects for volumetric MRI. So in terms of mode of delivery, and also things like if you get enlargement in the ventricles. I'm going to come back to that in a moment. Finally, how much change in your volumetric MRI biomarker is required to be clinically meaningful? So firstly, let's look at the Unicure trial as an example of how treatment side effects may confound therapeutic effects. So we're all familiar with this trial. So the gene therapy is injected directly into the caudate and putamen. But there's really a sparsity of data on how that gene therapy surgery will impact the volumetric MRI biomarkers in HD. So do we think the morphology of the caudate and putamen is altered by this drug administration process? Well, I found some data, gene therapy data, in a different disease. So this is late infantile Batten's disease. And you can see these are T2 scans. We've got immediately post-op. We've got 6 months later, 12 months later, and then 18 months later. And the yellow arrows point to MRI abnormalities localized at the sites of the catheter tips. So this is where the highest concentration of the administered vector is. So it seems that in this case, we have a prolonged MRI signal related to the surgery, Whether that's also the case for the HD trials, I don't know, but I would suggest that good communication and sharing of trial findings would be really critical in moving forward. It may well be that caudate and putamen biomarkers are not suitable for assessing the effect of the intervention on slowing atrophy in these trials, and we should start looking forwards to alternative measures. I'm going to shift to the tominersen ASO study, Generation HD1. Again, we're all very familiar with the results of this study. So we saw a sort of dose-dependent increase in ventricular volume in this study. However, there was an absence of increased whole brain atrophy rates. So this is a dissociation from the natural history data, where we often see very close links between whole brain atrophy and ventricular expansion, and often people think they're interchangeable. But I think this data is really important in showing that actually ventricular volume can change in a much more transient fashion, and whole brain volume is a more robust marker of global atrophy. So the ventricular volume is giving us information, but it's not showing us how the therapy is modifying whole brain atrophy. Another consideration is when you do get large expansion of the ventricles, there is some data that this can then result in unreliable volumetric MRI measurements. So that's another technical component to keep in mind. So finally, thinking about how much change in volumetric MRI biomarkers are required to be clinically meaningful. So I've chosen the Legato HD study as an example here. So a phase 2 study of laquinimod in Manifest HD. I think this was published recently, but we're probably all very aware of the study. So the primary endpoint, which was change in UHDRS TMS over 52 weeks, was not met. However, the secondary endpoint, which was change from baseline in caudate volume, was significantly reduced compared with placebo. And there were also 3 exploratory imaging endpoints in that study, so change in whole brain volume, ventricular volume, and white matter volume. And they were all consistent with the caudate finding. So I guess there are a few explanations. There's some train of thought that people think possibly a change from 4.8% to 3% in caudate reduction, in caudate volume change. Is that enough to be clinically meaningful? Or maybe the length of the trial wasn't long enough to detect an effect in TMS. I think it's difficult because the reason why we're developing biomarkers is just the clinical rating scales lack sensitivity, and then we end up in a situation like this where the biomarker looks promising, but the clinical rating scale doesn't show a change. So it's a sort of a catch-22 scenario. And I really enjoyed listening to Christina's talk earlier when she considered or maybe posed the question that maybe we need to start flipping endpoints, considering looking at maybe caudate volume change as a primary and clinical endpoint as a secondary, and having our trials over longer intervals. Maybe we need 2-3 years, etc. I hope I haven't misquoted you, Christina. So caudate atrophy in particular does look really promising, but what about defining and addressing the needs for regulatory endorsement? Well, the Biomarker Working Group of the HD-RSC is an initiative driven by CHDI and also the Critical Path Institute, and they are doing a lot of work in this field. And I'd like to point you to these 2 publications that have come out in the last couple of years. One's an evidentiary review, and another is recommendations to optimize the use of MRI in HD trials. They're both really good papers, and I recommend them. So I'm just going to wrap up now sharing some data from the HD young adult study. I don't know. Are we good to continue? Okay. So the HD young adult study, we performed really deep phenotyping on a cohort of individuals who were a long way from predicted clinical motor onset. One of the aims of this study was to develop biomarkers that may be able to facilitate the design of future preventative trials. So in brief, we recruited 64 HD gene expanded individuals who were 24 years from predicted clinical motor onset, and they were all young adults. We got some matched controls, and so far we've assessed them at 2 time points with 4.5 years in between. We've done a really in-depth cognitive and neuropsychiatric battery, which takes about 2 hours, and it's tricky. We've also looked at biofluid biomarkers, both in the CSF and the blood. And we performed a wide range of neuroimaging, including volumetric MRI, which again has ended up being the most sensitive. So at baseline, 81% of our cohort were HD-ISS stage 0, 17% was stage 1, and we just had one individual who was stage 2. Over the 4.5-year interval, we had about 18% progress from stage 0 to stage 1, no one progressed from stage 1 to 2, and no one regressed. I'd just like to show you a little bit of imaging data now. So this is an example of one of our participants who was stage 0 at baseline and then transitioned to stage 1 during the study. So this is their baseline MRI scan. And this is their repeat scan about 4.5 years later. So I just moved between the two. The brains are spatially aligned, so you can actually visually see the differences in the brain for this individual. So we use a technique called the brain boundary shift integral to quantify this whole brain change, and that's the overlay for that. We can also localize the change using a different technique. So this color overlay indicates where there's been change. So the cold colors show you contraction or atrophy, and the warmer colors are where there's been expansion. So in this individual, it's clear that a lot of the changes are occurring in the caudate, the putamen, and the peristriatal white matter. We use a whole range of different image analysis tools to get the most robust and reliable results in terms of annualized volume change. Here are some of the results for this participant. So everything is slightly higher than you'd expect for an age matched for a young person, particularly the putamen and the caudate. So if we look at this data at the group level, we can see that in HD-ISS 0 and 1, atrophy rates are already significantly elevated, and we were not expecting to see this. So here we can see the putamen and the caudate, gray is our control data, and pink is our HD gene expanded data, and there are already marked changes, particularly in these two local regions, consistent with what we see later on in the disease. So if we look at our HD group in a little bit more detail, so here I've plotted caudate volume and putamen volume against CAP 100 score. So our HD-ISS stage 0 are in orange, stage 1 in red, stage 2 in blue. And the black lines just indicate the longitudinal data. So we can see that almost everyone is losing volume, even those who are stage 0, which is basically the top of each graph, or those with a particularly low CAP 100 score. So what's also quite interesting is that change in caudate and putamen volume was strongly associated with change in CSF NFL and change in PENK, even after controlling for age and CAG. And you have to bear in mind that these changes are occurring in the absence of any clinical decline, cognitive decline, or neuropsychiatric decline. So we hope this data will be informative in helping to sort of generate a biomarker panel for future preventative trials in HD, which is our ultimate hope. I'd like to thank our HD Yaz participants who are lovely and we've got to know really well during the course of the study. Also, thank you to our funders and collaborators. Thanks for listening.
For a wonderful talk. I always feel like a good sign of good talks are when everybody's got their phones up, taking pictures of all the slides. Lauren, in one place where you were rapidly going through, I saw a lot of people switch to video. They couldn't take pictures fast enough, and they just said, I'm just going to video Lauren's talk. We'll we'll open it up to the floor to questions. I'll kick us off with something, I love this conversation about that biomarkers and clinical trial endpoints, it's contiguous, right? Biomarkers are clinical trial endpoints. And as you mentioned, Christina's throwing this out there, and I think a lot of the companies are thinking about this, flipping to biomarkers as primary endpoints when our previous primary endpoints have been mostly clinical. So what I'd like, the question then is for each of you guys, what do you think about the readiness of imaging and fluid biomarkers to be primary endpoints? And then Tarina, I know you don't speak for FDA, but FDA's receptiveness for having clinical trial endpoints, biomarkers as primary endpoints for clinical trials.
Yeah, it's a tricky thing to say, kind of a blanket, because obviously as we try to look, every modality is different, every context. Obviously, I probably have to vote for NFL because that's my protein. But I think we have to seriously consider how long it might take to change that. And it was touched on yesterday, but I think any of the drugs could work, and I think I hope a lot of the programs will be successful. But I think my expectations of how much impact they have is maybe more conservative than others, and I think we need to think of these maybe first line of drugs going to be a small dent in the kind of progression of HD, so we need to be prepared for very maybe small changes in the outcome measures. So yeah, I think having trials that go to 3 years would be really helpful.
Right, and I think and again, I'm not speaking for the FDA, but in the engagements that we have with them, the conversation is always an important back and forth of giving them needing to see data and evidence in order to inform their decisions, and then the community reasonably asking for where the line is for the opportunity for what we call regulatory flexibility, but I prefer to call regulatory optimization, because the whole point is to not lower the bar for rigor. The importance of safety and efficacy is really high, but as we all know, the complexity of Huntington's disease, rare diseases being limited by the number of participants, this is where I think there's openness. There's a general receptiveness that the FDA has for acknowledging the challenges in the HD space and the need for regulatory optimization, but that is an iterative bilateral process of information, data, evidence, conversations. What really is key is when exploratory and secondary endpoints are included in clinical trials that then develop really rich data that can feed that information process faster. So I think that's really important.
So I think there is a potential role for caudate volume change as a primary endpoint at some point. I think what I was trying to get across in my talk is that I don't think it's a one-size-fits-all solution. And I think we really need to be thinking about, the mode of delivery of the therapy. We need to be thinking about the mechanism of action. There are lots of different factors, so we can't really, I think, you need to select particularly your imaging endpoints based on, the design of your trial and what type of therapeutic you have. But I think in terms of sensitivity, it really is very impressive longitudinally, and it's you can measure it objectively, reliably, and it's just fundamental to the progression of the disease.
Addis, you want to start?
Sure. Thank you all for such a great presentation. Addis Mendizabal, UCLA. One of the questions I have for all three of you has to do with the generalizability of these biomarkers in diverse populations in a twofold way, right? Like if we're looking at the patients in the clinic and then we're thinking of developing a biomarker based on what we're seeing in the clinic, how diverse are any sort of the data sets that we're using to identify different biomarkers? And then also the other way around, because if we're talking about looking at the endpoints that clinical trials are using, And we already know that there's a huge lack of diversity in the clinical trials, and we're seeing not only racial and ethnic diversity, lack of diversity, but also we're seeing more highly educated patients with more resources that are part of those trials. So if we're developing biomarker endpoints based on that, then we're also having another selection bias. So I would love to hear a little bit of your thoughts on that, how to address it, mitigate it, and then also if you have experience with some of these data sets, talking a little bit more about the diversity in them.
I think it's an excellent point, and I think it's a big problem. I think in our observational research, we recruit people who, A, have decided to have the predictive genetic test for HD, and secondly, have decided to embark on research. So they typically, they're very well educated and they have an IQ higher than average. So I agree with you, I think it is a problem. We try to help with socioeconomic diversity or inclusion by paying for everyone's travel up front. We pay and book their hotel and accommodation. We write them sort of letters, to help them just get the time off work without having to lose out on it. So we do what we can, but I think it's a problem.
Yeah, I think it's a huge problem, Alice. And one I think with the retrospective data we have, we're limited to. But it's something I'm particularly passionate about, and why I've been, one of the reasons why I think be able to have a remote collection method will be particularly useful. The nice thing about a blood biomarker, it's easy accessible, and the fact that NFL is somehow really stable and doesn't get affected by delayed process. And my aim is to make that a way in to expand into these kind of deserts of places where there isn't access to specialist centres. But there is still an issue about engagement of populations that don't engage with research. So it's not even the fact that geographically you might not have access, but there's also, the burdens of cultural and social impacts. So that's something we need to work on more, I think, as a community with the patient organizations, but at least having something that people can opt into at home. So it's I'm hoping to apply this at-home blood collection to Join HD, which is a registry people with JHD and their caregivers can self-enroll anywhere in the world, so it's global. And the long-term aim is that they can opt in to also doing a blood collection that will be sent to them in their own home. They collect it themselves, send it, so they have control, and if they want to take part. But again, that doesn't, people still need to know about it and still need to feel comfortable, and that's a wider systemic problem in kind of medical inequality.
And then just some punctuation, because everything that they said is what I completely agree with, and absolutely acknowledging the importance of the question and the gap that the field faces. And this is not a unique problem to Huntington's disease. It's obviously something that I think will benefit from other groups, other disease areas, other collaborators to really work together to try to rise all boats. One thing or some things that may inform that process is, again, the fact that the FDA acknowledges the importance and the danger of not having diversity represented in clinical trials. So there is a recent guidance that has been put out to try to help sponsors find ways to increase diversity and inclusion. And then recently, the National Academies of Sciences, Engineering, and Medicine published, it was actually an interesting resource that's kind of provocative in questioning how we've understood and represent these key questions so far in medicine and research, and whether it needs a holistic rethink. So obviously this does not solve the problem quickly, but I think, as I mentioned, since it's pervasive across multiple diseases, I think this is an opportunity to reach across other groups. And then the last thought that I have coming to NFL, what is, and we all know it's Lauren's favorite, but what I think is important there is, while we all acknowledge that it's a very specific and sensitive marker, but it's not disease specific, it's neuron damage specific. But that actually provides potentially an opportunity, coming again to this whole reaching across and collaborating with other stakeholders. Since so many different disease areas are looking at neurofilament light, there is an opportunity. It has not been actualized or realized or implemented, but an opportunity to make normative databases, because so many diseases in different ages and demographics and geographies are looking at the same marker. if in a perfect world where everyone shared data, there's an opportunity for getting information out of normative databases in that regard. Call to action.
Sorry, Nick McFarland, University of Florida. I'll just make more of a comment, but I think when we think of biomarkers, we often do focus on biofluids and imaging, which are very powerful. And I think if we could use it as surrogates, I think that's sort of a goal, but I think we're also moving towards earlier parts of disease. I think I'm impressed with the data from 0 to stage 0 to 1 imaging. That's huge. If you can detect a 1% change in brain volume, that's amazing and shocking. But I think what I'm pointing at is I think we need to not forget about the need that maybe challenge the rest of us, that we need some earlier clinical biomarkers or even some quantitative measure biomarkers to match this, to go to the FDA to say these things match. So, I mean, I think Karen McDonald mentioned things earlier today and talked about kind of better sensitive ways of, looking at non-motor symptoms that might occur really early on in disease, maybe even creeping into stage 1 that we haven't detected before. So I'm just thinking of, we can't do these in isolation. And if we have to think on our side from the clinical side and maybe some measures,
Oops.
I was just going to maybe follow up with, at least in HD young adults, like there were a lot of more advanced psychometric tools that were applied. I think it's just a kind of crazy phenomenon, the fact that it takes so long for people that carry the Huntington's gene to have these biomarker changes for them actually to develop symptoms. I do think there's, avenues like the digital motor score and digital measures that definitely need to be looked at, but I still think there's going to be a 10, 15-year difference in kind of when we see changes in these really earliest biomarkers, at least for caudate volume and NFL changes.
Thank you for your wonderful presentations. I was thinking about this, NFL and caudate volume, they seem on their face very good as candidate biomarkers for assessing treatment response. Their sensitivity to change is indisputable, their association with disease status indisputable. But then comes the question of, if you're actually going to try and flip it to be a primary, as Christina has suggested, what are credible not effect size change estimates over time, but treatment effect size assumptions that would give a sponsor the courage to actually pick it? And it leads me to wonder, we've seen, I guess, for the first couple of times, you mentioned the Legato findings, WAVE showed a caudate effect. What are those treatment effect sizes, actually? Do we believe them? Do we find them credible? Because I think for a sponsor to pick it, you before you get into like a 3-year trial, without really knowing, what is that % treatment effect size going to be? It would seem risky to choose your sample size and actually be powered to show an effect. So I don't know how do you see that issue? How do you see us getting to a point where we can turn an abstract conversation into, action for sponsors?
I think at least for NFL, we're not there yet. All of the trials, the more advanced trials today and the data that we're basing NFL responses on have had, unfortunately, adverse effects and safety in NFL. So until we have trials that don't cause transient increases, it's hard to then look at NFL as an outcome measure, as a surrogate or for benefit, because it has to come back to normal and then go down. So, and as we've seen with other degenerative diseases, it's quite complicated. So I think we need more data with NFL. It's not there yet to be a primary endpoint for sure, but I think it has a lot of value in terms of stratification at this stage, and once we have more data across the HDISS.
So one of the things that I'm thinking about in this regard, and again, it comes back to really trying to understand where there's opportunities for regulatory flexibility, because that's kind of what we're also talking about here, is what evidence do they need to see, what would de-risk a sponsor to go into this space. There are a couple of areas, Christina talked about some areas where we can share learnings earlier from the Friedreich's ataxia approval and from the tofersen approval for SOD1 ALS. There is also a precedent for a biomarker that was used through the Polycystic Kidney Disease Outcome Consortium at the Critical Path Institute. Clearly, we're not talking about kidneys, but this was a rare degenerative kidney disease that used volumetric MRI of kidney that was correlated to glomerular filtration rate, and they could show through modeling and through data that it was important in the pathology and progression. And then that got approved by the FDA as a surrogate under the very specific context that a sponsor could use it as their primary endpoint for consideration for accelerated approval, but they had to come to the FDA and discuss the data at 1 year in an interim analysis to see what the change on the biomarker looked like and agree to either continue or discontinue the study based on that change. I'm not implying that this is a plug-and-play process that we can do in this space, but it demonstrates another rare disease imaging biomarker and the FDA bringing to bear some flexibility.
Diana Rosas from MGH. So great talks. I think this is incredibly important work that you guys are doing. I had one comment first to Nicola. I think that you kind of played a little bit the importance of the quality of the data acquisition. As you know, the vendor, the head coil, field strength, even positioning within the head coil are all things that are actually going to modify your acquisition. And then, software upgrades in the data acquisition over time has always been a problem with every imaging center that exists, right? So just to put a plug into how important it is for people to understand the rigor that's required to collect the best data set, because the best data set at baseline is super critical. I have just a question. One of the things that I think was missing and I think was brought up at the first talk was the importance of retest reliability, because I think, you saw the stuff that's after that Lauren sort of after that you heard Nicola after 4.5 years, but when we're looking at potentially trying to get more efficient trials, the goal is for these biomarkers to actually be better at predicting outcomes than we have at a clinical trial. So if we have to wait 3 years, well, you can just use like a TFC or a time to conversion potentially, right? So do you have retest reliability that actually looks at within 3 months or 6 months within the same participant? Because the stuff that I've seen sometimes is the heads will get bigger, they'll get smaller, and then there's a lot of variability that can be induced, and then thoughts about how we could actually reduce that.
Thanks, Diana. Just 100% agree with you that the quality of the raw data is absolutely imperative. And in multi-site clinical trials, you need a really good central imaging facility to lead on that, and a facility that has, or an imaging CRO that has experience with Huntington's disease and knows how to maximize the quality of the raw data, because, that's everything. You can't correct. If your baseline data are not good, your imaging will not be reliable. And a lot of the results that I've shown, particularly for the YAS study, we've got to remember that's a single site, highly controlled environment. So the noise is very small in that situation, and it's kind of like a gold standard approach. And even with the best planning, there will be more noise in a multi-site trial. Absolutely. So I agree with you. So the YAS data, the interval is quite long, being at 4.5 years. I think we didn't expect to see as much as we have seen. So we're currently partway through a third visit, and the interval between visit 2 and 3 is much shorter. So we'll be able to get, we'll have 3 data points for each individual. We'll be able to look at trajectories of change, do more advanced modeling, so that will be informative. One thing I didn't show was the control data over time, mainly because I didn't want to plot the data by age, because I don't want anyone to be identifiable. But I do have those graphs, and I'm happily show you. And we actually see that the control change over time for the caudate and the volume and all of our imaging metrics, volumetric measures, basically it's a flat line. So we're not seeing, these are very young individuals. They're between 18 and 35 years old. So, pardon? I'm one of the lines. Oh, yes. Lauren is in our study as well. So yeah, the volumetric imaging is very static in the control group. And we have done retest reliability. So we've got We've done back-to-back imaging previously. Also, at this time point, this is a slight deviation, but we're collecting both 3 Tesla MRI and 7 Tesla MRI on the same people. So we've got paired data. So we're looking at differences across scanner field strengths as well. So yeah, we obsess over the technical details. So it's very important. Thank you for raising it.
Just to add on kind of, it's something I'm interested in and my team is developing ways we can harmonize retrospective data that has been collected on various methods. And there are new methods out there that like combat or more advanced measurements that kind of try and control for batch effects and things like that. So we are looking at ways to do that as well. But again, I think nothing's better than actually standardizing from prospectively.
We can take one more. We can do
one
one
more?
more quick question.
I have the mic.
Okay. Christina, go ahead.
So I was here holding a comment question to Nikki regarding the Legato trial, because the Legato trial is published in Lancet Neurology. It has the protocol as a supplement. And if you go to the protocol, there are a couple of details that are super important. And I'm insisting on this because all of us are looking for a clinical trial, intervention trial, that has MRI data that actually moves. And when we think about it, the only one that we have is Legato. In the Legato, there are a couple of problems. One of them is, first, the MRI was defined as a co-primary endpoint. This is not a problem. It would be a big asset. But the fact is that when we look to the data and we look to the protocol, in the protocol, there was a planned analysis for the MRI. And the change that is planned there predicts that the placebo arm would have a certain atrophy. So during the duration of the trial, the placebo arm should have gone through a certain level of loss that is in the protocol. And then what we see in the reality is that the placebo arm lost almost double of what was planned in the protocol. So what this means is that the placebo was the group that changed more than it was planned, while the intervention arm almost followed what was planned for placebo. So does not seem to be an effect in the treatment arm, but rather unusual let's say, loss in the placebo arm. So this speculation about what would be an effect size in Legato is less appropriate because of this mismatch between what was the supposed loss in placebo and what actually happened. So this is one of the problems with this data set. These things happen in reality. But it's why this data set might not be so helpful. And I would say, let's say my next comment was regarding what Scott asked about how much a biomarker needs to change to be quote-unquote clinical relevant. We don't know, indeed, but the fact is that almost across all diseases where biomarkers are used, you typically need a very large effect in the biomarker to have an effect in the clinical outcome. But I would say to companies that they are estimating changes in clinical outcomes, and they are betting more than having certainties. So they should bet, because we need to generate data. They should bet on the biomarker change. Probably the bet will be a large change, which the trial will be smaller. And then we have to acquire data. So, and while I have the microphone, I make a last comment regarding diversity. Diversity is really a very important topic, and when we are planning trials and other studies. But I would say we need to decide what is the topic that we want to address by increasing the diversity in our cohorts. And I truly think it's important, and we are doing a lot of effort in Enroll. But when it comes to the technical aspects of MRI and the technical aspects of NFL, in these particular cases, these methodologies are transversal to a number of diseases. And there are consortiums that are aggregating data from different diseases, like the Enigma Consortium for Imaging, and there is efforts in NFL. So pulling together data from many diseases, there is a lot of diversity in those, much more diversity than we have. So we can have some glints, some insights about the impact of different ethnicities on the technical aspects of these biomarkers.
Thank you for your comments. We do have to end there, unfortunately, so that we can stay on time, but we do appreciate your feedback.
Am I just able to
Yep, you have 1
respond?
minute. Go right ahead, and then we're going to continue on.
Okay, I've got 1 minute. Just in terms of the Legato HD study, I think that I would argue that it is valuable to look at the difference between the placebo group and the treated group. And I completely agree with you, Christina, that the placebo group had a higher atrophy rates than we've seen in natural history studies. But so much of that is dependent on the image analysis tools and how they're implemented. And I would argue that maybe it would be worth reanalyzing that data using different image analysis tools and just to see what the results are, whether the placebo group is then more in line with what we see from natural history studies. It might be interesting to look at.
Wonderful. Thank you all who participated. Thank you to our speakers. Thank you to Dr. Sung. Appreciate that.